Hello there, this is Dr. Alleles. I'm going to go through the the genetic side of the lessons that we've been doing in school. So if you've missed it, then they should hopefully be helpful. I didn't give any handouts, so feel free to make your own notes, etc. So yeah, the aim of what we're doing, we're going to check, really just to check your understanding of some difficult topics. Obviously, you can't ask any questions, but you can email them to me, a.ellis@crgs.org.uk, Ellis, E-L-L-I-S. Yeah, so let's have a look. So variation, there's three reasons or three measures of variation. So yeah, these big questions. Why do muscle cells vary from nerve cells? So in an individual, why cells vary? So just to help us get there, what do genes code for? Well, they code for proteins. And are all the different cell types genetically identical? Yes, they are, because they've come from that original sperm met egg. Then that makes a zygote, a stem cell, and then from that point on, the only differences between the cells of our body is genes have either been turned on or turned off. Something we do in year 13 around January time. We look at cell specialization. So number one, we've not been bothered about why we vary inside our bodies, why our cells vary. Number two, we have been concerned with 2 and 3. So why do members of the same species differ? Well, do members of the same species have the same genes? Yes, they do. They differ because they have different alleles of genes. Okay. Yeah, so I can say, yeah, they do have the same genes, assuming they're the same sex. The reason why they differ, so yeah, genotype, because they have different alleles of genes, and the environment, really important. There's an important N in environment when you're spelling it, and also the fact that the environment can cause a change to a phenotype, yeah, an observed characteristic. So we vary because of our genes, or alleles of genes more precisely, and the how the environment affects it. So I get more freckles in the summer because of the UV light. Number three, why do members of different species differ? Well, what's the definition of a species? They can breed and produce fertile offspring. And what must gametes have to be compatible? Well, they need to have the same kind of like order and size of chromosomes. So yeah, the same genes need to be on the same chromosomes. When sperm meets egg, the chromosomes come together, the pairs are kind of matched together to check that they are the same thing, and the only ones that are allowed to differ are the sex chromosomes, the X and the Y, and that's not a problem if you're female because there'll be 2 X chromosomes there, but it's not always as simple as that because there can be mutations that lead to differences of the sex chromosomes that we meet in year 13 a little bit. Okay, so taking from this slide is knowing that that there's variation, and variation doesn't mean one thing, it can mean different things at different levels. Okay, so we've been mainly working on number 2, why do members of different species differ, and then we looked at natural selection and directional selection and things like that, and then we've moved on to like different species in the environment and diversity indexes and things like that. So we're going to now go through how we get new alleles of genes, small scale mutations. So I've got my DNA code, TACGC GAAC, and we're going to do the mRNA transcript. Well, T, that will complementary base pair with U, or it won't, it's A. A goes with U. I always get that wrong, I need to remember that. Yeah, so thymine is replaced by uracil in mRNA. So I can pop those in. Just take care, because one little relaxation and you'll get it wrong like I did. Then we've got here, it says the types of small scale mutations. These are the ones that we need to know: addition, deletion, substitution. So this is a normal protein. Look at its shape. This is an abnormal protein due to changes to the DNA, and as you can see, it's got the same start, but then it's longer. It's like, how can you get a longer protein? Okay, this is where a lot of people kind of don't understand about DNA and mutations and how ribosomes work. So here, just at the top, I put a piece of mRNA. The green shows me the start codon. I don't know why I said codon poshly, but never mind. And red is the stop codon. You can see this overhang here. A ribosome will kind of hook up onto the start, move along, and then it will only start translation when it gets to the start codon. So it'll translate between the green and the red, and then it hops off at the red. So if we lost the stop codon, then that would cause it to just carry on going, and it would just drop off the end and make a larger protein. And that could be due to anything. It can be due to an addition or a deletion. That would cause a frame shift, or they'd cause a frame shift. Or it could just be a substitution. Anything that will get rid of a stop codon. Yeah, and there's 3 different sequences for stop codons. I'll show you on the next slide. This other one here, this is a smaller protein. Okay, so how can I get a smaller protein? Well, back to this diagram, if I shuffle that red inwards, I'll end up with a smaller protein. And that's therefore this is like the opposite of the first one. You can get a premature stop codon. So you can lose a stop codon, or you can get a premature stop codon. Again, that could be due to addition, deletion, or substitution. There'd have to be just the right bases there that just happen to cause a premature stop codon. Next one, this is subtle. Can you spot the difference? This is it's like rounded there and it's wobbly there, but the rest is the same, isn't it? So yeah, there's a subtle difference. How can I get a subtle difference with a mutation? By addition, deletion, and substitution. Well, if it's just one base, the only way I can do it with one base is by substitution. So if there's a small subtle change, it's generally by substitution. Therefore, that codes for just one different amino acid. So the R group of that amino acid will bind differently to the ones around it, or the one around it, and therefore it'll cause a slightly different tertiary structure in the protein. So yeah, small changes, think substitution. This last one is vastly different. Yeah, it looks nothing like this first one. So it's vastly different. And if it's vastly different, that's likely to be due to a deletion or an addition. Again, it's the same, similar length, but yeah, it's vastly different. It's even got it's got a different ending and start. So yeah, that's where if I delete a base out, that causes a frame shift because the genetic code is read in 3s, and therefore if you lift out one of the letters, all the codons after that point will be read in a different order, or read, one base shimmied along, and therefore you get different amino acids that bond differently, and therefore you get a different shape of protein. So let's try this one. Can we describe the mutation that would result in the protein being identical apart from missing one amino acid? So the mutations. Mutations. So how can I get mutations that would result in it being identical apart from one missing amino acid? So not a changed amino acid, so it can't be substitution because that'll just change it from one to the other. Again, I guess you could say that the last codon before the stop codon gets changed into a stop codon. You can do, but it's probably more likely to be a consecutive 3 base deletion that results in a codon being removed. So if I lost GCG from this, then I would lose one amino acid. 3 bases code for an amino acid. This is a triplet of bases. In mRNA, it's a codon. So you push again. Yeah, so it's a deletion of 3 consecutive bases in DNA to remove one entire codon in mRNA. And there's other ones that it could possibly be, but that's the most likely. So here's our codon map. And you read this I've put the mRNA transcript here. So this is an mRNA codon map. Well, codon is a word that goes with mRNA only. I know it's mRNA as well because it's got U's in it, not T's, so it's not DNA. So my first codon is AUG. We read them in 3s. And AUG starts here with A, go to U, go to G. That will give me methionine. That's given its first three letters. So all these are amino acids on the outside, and they're all known by their first 3 letters, with the first one capitalized. You don't need to know any of these. Here it's telling us about different properties of them as well, which isn't necessary. Then CGC, so can you work out that? CG, and then any other letters, that's arginine, ARG. And then UUG, UUG is leucine. Yes, that's how we read these codon maps. Sometimes they're more tabular than round, but these round ones are nicer, I always think. So can we describe a mutation to the DNA that will result in the primary sequence methionine, arginine, methionine? So I'm going to keep methionine. We're going to keep arginine, but I'm going to change that to methionine. Well, methionine, so I've already got it. It's AUG. Yeah, so I need to swap the U for an A. Is that it? So why does this get 0 marks? The first base of the third codon, yeah, here it is, was changed to A to make the codon AUG. Well, that sounds perfectly correct, doesn't it? But it gets 0 marks because I've not described the mutation. I've not said what kind of mutation it is. Is it addition, deletion, or substitution? And I've talked about changing the mRNA, not the DNA. You see that? Again, really favourite question is this. So for 2 marks, adenine has been substituted for thymine. Substituted, I've named the mutation. In the DNA, it'd be an A that codes for the U. So the A must have been changed to a T, because that way, if it's T here, T in the DNA, I'd get an A in the mRNA. Again, really common question. Most people get it wrong. Get 0 marks. You can at least get 1 mark for naming the mutation. Okay. The next bit is yeah, just be on your guard, because this otherwise is too easy for 2 marks. Number 3, page 3, which is that was like page 2A. Insulin, a protein that is made of 51, and should say is a protein there. Sorry. So what is the minimum number of bases that code for 51 amino acids? Yeah, so how many bases? So that's 51, and 3 bases code for an amino acid. So it is 150, 153. Yep, I'm reaching for my calculator. I know 153, correct. Why might the actual number be 156? Well, oh yeah, there's a stop codon, isn't there? And the stop codon doesn't code for an amino acid. If we just zoom back quickly, there's a stop codon here. It can be UAA, UAG, or UGA. Any of those, they code for a stop. A stop isn't an amino acid, it just means stop. So yeah, that can be explained by the stop codon. Yeah, 156 would mean I'd have 51 amino acids, and the last three bases would code for a stop codon. Would be a stop codon, yeah. Why might the actual number be 159? So that's adding 3 more on. Well, that might be the start codon. Yeah, the start codon calls for methionine, as we showed previously. However, sometimes that's removed because I don't want to start every single protein with the amino acid methionine. So often when proteins are made, you lose the first amino acid because it just happened to be coded for by the start codon. So yeah, the start and stop codon have been included in C. The actual human insulin gene is 1,431 base pairs in length. How can this be explained? So the gene's a lot longer than 159, isn't it? Even the biggest I've got to. So how can that be explained? Remember introns and exons. Yeah, so introns have been spliced, removed, and the introns have been removed and the exons have been spliced together to form mRNA. So I'm going to talk about pre-mRNA. In that the pre-mRNA would have been that long, but then the introns were removed and the exons joined. And then why is it better to say base pairs for length of a gene rather than bases long? Well, bases long is vague, isn't it? If I'm talking about a gene, genes are double-stranded DNA. And then if I talk about how many bases long it is, am I talking about just one strand of DNA? Am I saying it's 100 bases long? Or am I counting both of them when I say that? It would lead to misinterpretation. Some people might think it's 100 actually length-wise, or some might say it's 50 because there's 2 bases for every kind of like unit length of DNA. So yeah, it's better to talk about base pairs. We measure DNA tends to be in like kbp, kilobase pairs. How many thousand base pairs is it? Is that gene? It's 14 kilobase pairs. Okay, so yeah, the actual way of saying how long it is, not how many bases. Expect it in exam to talk about base pairs if it's talking about DNA. Right, so how does a mutation result in a non-functional enzyme? So you're going to have a go at this. So pause it and have a go. See if you can get the 6 marking points. I'm just going to carry on now. So when I teach this at GCSE, it's very similar. I talk about sequence, sequence shape. So I just add 3 more bits of information. So there's a change in sequence to basis of the gene in the DNA. That's what a mutation is. It's a change in the sequence or amount of DNA in a cell. And if this is talking about an enzyme, we're talking about a gene, therefore we're talking about a change to its sequence. Therefore, there's a change in sequence of codons in the mRNA. Okay, so when it does transcription, it's going to change. There's going to be that change in the DNA is going to be reflected in the mRNA. Therefore, different tRNA anticodons bind to mRNA codons. Okay, I always try and get that into my answer. So I'm talking about translation now. Mentioned that the tRNA, that's the thing that determines the amino acid. So 4 then says ribosomes join amino acids in a different sequence because of number 3. Therefore, or there's a different primary structure, I could say. But I did have to say the bit before the hash. Therefore, we get different bonding between amino acids due to different R groups of the amino acids. Remember, the R groups of the amino acids determine what kind of secondary bonds it forms, secondary and tertiary bonds. Therefore, that gives me a different tertiary structure of enzyme. Have I answered the question? No, I've not. How do I get a non-functional enzyme? The active site is no longer complementary to its substrate, or can't form enzyme substrate complexes. So there's 7 ways of getting the 6 marks. So you could easily miss out number 2, I guess. But yeah, don't forget about it's the tRNAs that are determining which amino acids are going to be joined because they're the ones carrying the amino acid. And it's the anticodon-codon pairing that determines which tRNA binds in the first place. So yeah, well done if you've got 6 marks. If you haven't got 6 marks, learn it. I think this is something that my old maths teacher used to call book work. There's plenty of hard stuff in maths, but learn your book work. Yeah, stuff that we can just regurgitate in an exam or have it in your head. Sequence shape. And they often ask about enzymes because then they can ask about your knowledge of active sites and ES complexes. So why might a mutation within the amylase gene of a cell have no effect on the functioning of the cell? So how can I have a mutation and I still get functional amylase? Well, no, it's not saying that, is it? Functioning of the cell. Well, the gene might not be turned on. Back to that first slide. Yeah, that if the cell doesn't, if its job isn't to make amylase, then it doesn't matter if it mutates the amylase gene. Yeah, the gene is just not expressed. It's turned off. It's wrapped around histones. Genes are in pairs, so still has one functional copy of the amylase gene. So even if it does need to make amylase, well, genes are on homologous chromosomes. They're in pairs, one inherited from each parent. If one's got a mutation, you've still got another functioning one on the other copy. You can say that the mutation takes place within an intron of the gene that's removed in splicing. It could be a substitution mutation that results in a codon that codes for the same amino acid. We call that the degenerate code. Again, remember that codon map a couple of slides ago? There was 4 ways of coding for one of the amino acids. I can't remember what it was now. I think some amino acids, there's 6 ways of coding for it. So yeah, there's many ways of coding for the same one, and that's where degenerate comes from. It's a mathematical term for numerous solutions to a coding system. Or we could even say that the mutation in the DNA that codes for the mRNA before the start or after the stop codon. Remember, it only translates between those 2 points. So it might be either side of them. Again, I won't particularly put that one because it's a bit obscure. But yeah, it's amazing that that people don't think of the first one. They think of every cell doing everything, and all cells don't do everything. We can even say, that it's corrected. And the mutations, if I change one base on one strand, that's going to mean it's not longer complementary to the other strand, and therefore we have repair mechanisms that come in. They use the non-damaged strand to rebuild the normal strand, the complementary strand. And these, 2 types of kind of repair mechanism that we have that we don't learn about, but not all mutations will have an effect. That's why diseases due to mutation are incredibly rare, because we have a repair mechanism. Right. let's look at cell division. So we're on to cell division in eukaryotes. So there's two types of cell division in eukaryotes. There's mitosis and meiosis. If it was prokaryotes, it'd just say binary fission here, and we'd have done with it. So where does mitosis take place in an adult male? Where in the body? And this is where at GCSE people say everywhere. Not everywhere. It's anywhere that's growing or repairing. Skin cells, muscle cells, bone marrow, that's where mitosis takes place. Yeah, go to the gym, which I can't do anymore. Not that I went anywhere. But if I could pump some iron and grow some muscles by stimulating muscle cell division. Meiosis, where does that take place? Well, that takes place in the testes. It doesn't take place in sperm cells. Sperm cells are the product. Sperm cells are haploid cells. They are the product of meiosis. So there's stem cells within the testes that are undergoing meiosis, and the product of that is or are the sperm cells. So yeah, testes is where meiosis takes place. In females, you say ovaries, but it's more awkward because you can't. Meiosis doesn't take place in adult females because meiosis happened when you were a fetus. You used to be an egg cell inside your grandmother. That's frightening. As in, when you were the egg cell that became you was already in your mother when she was a fetus, and when she was a fetus, she was inside her mother, your maternal grandmother. Frightening. Anyway. Why is mitosis needed? As I said before, growth and repair of tissue. Why is meiosis needed? For a few reasons, but crucially, to make haploid gametes, so that the diploid number can be restored at fertilization. And we do it to get variation in offspring. So the only way we can get variation in offspring is to combine genetics of two individuals. And therefore, I don't want to double the amount every time, so we have to halve it first, so that we can restore the normal number of chromosomes. So yeah, you should be using haploid and diploid at A level. And if a diploid number for this organism is 20, how many chromosomes in cells produced? Well, it would be 20, 10 pairs. So the diploid number is just the number of chromosomes in a normal body cell. Okay, so the diploid number for humans is 46. Okay, so meiosis, the number would be 10, one of each pair. Okay, hopefully that's making sense. Again, this is hopefully where you're kind of like nodding along saying, yes, it makes sense, or no, this doesn't make sense, in which case you can do something before September. Catch up. Topic 4, why are these incorrect? So sperm cells carry out meiosis and are therefore all genetically different. Yeah, sperm cells are the product of meiosis. So A is wrong for that. A homologous pair of chromosomes are held together by their centromeres. No, they are not. Homologous chromosomes are never joined together by centromeres. They are separate chromosomes, aren't they? One inherited from each parent. When a chromosome copies, it has an identical strand. We then talk about them as having been made of chromatids. Then they are joined by their centromeres. So yeah, we need to make it really clear in your head what a homologous pair of chromosomes are. That's like my index fingers on each hand. They're like, I've got 2 copies of each index finger. They are homologous. If I bring in the other finger next to them, then I've like copied that finger kind of. I know it's a different finger, but kind of. Those 2 fingers on my right hand, they would be joined by a centromere, or by their centromeres. Each one's got a centromere joined together. Organisms mutate in order to survive. This is called adaptation. So can I now mutate if there's a change in the environment? Yeah, it's very, very hot today. You know, maybe having even less hair will protect me, or actually maybe more hair will protect me from UV. Who knows? But can I change now? No, I can't. Yeah, it's populations. Well, individual organisms can mutate, and then the best adapted will survive. It's not to survive. It's just if there's 1,000 organisms and they've all got differences about them, then one of them might be better adapted than others, and therefore that's going to have a greater success in that environment. So yeah, adaptation talks about a population changing. Yeah, how have giraffes adapted to their diet? Well, they've gained a longer neck. Yeah, they, as in the population, has over time. D, ribosomes read the mRNA code and ensure amino acids are attached in the correct order. Nope, they don't. It's all to do with the tRNAs and the mRNA interacting. So the tRNA's anticodon binds to a complementary codon on mRNA that positions an amino acid within the ribosome. The ribosome then just kind of blindly just joins whatever amino acid is there. It's not reading anything. It's not intelligent as a ribosome. It's just positioning itself over the mRNA, and it knows to start at the start codon and stop at the stop codon. Or if there's no stop codon, it carries on to the end and falls off, releasing the protein. And E, the genetic code is described as non-overlapping because genes occupy a specific locus on the DNA. No, non-overlapping is all to do with the fact that the DNA is read as codons. This is where the ribosome has a job. It advances 3 bases at a time. It moves on a codon. So it's read. It reads 3 bases, next 3 bases, next 3 bases. That's non-overlapping code. And yeah, genes can never overlap on a chromosome, but we never talk about them being non-overlapping because it wouldn't work otherwise. They have to have a part of the gene that is where the RNA polymerase binds, the promoter. It then reads that gene, making a pre-mRNA copy, and then it jumps off at the terminator sequence. Yeah, E's full of nonsense. They've confused DNA and mRNA. Good, right? We're gonna. So that's small scale mutations and a bit about evolution. Now we're going to do large scale mutations. So here's a diagram showing meiosis, and it splits into 2 parts. Does it say in the syllabus that you need to know all the stages of it? But yeah, this is where we've had a mutation called a non-disjunction, and you can see here the chromosomes have not done something. And the same thing here. So there's 2 places, either in the first stage of meiosis you can get this happening, or in the second bit of meiosis. And you can see here these cells are produced. Well, that's got 3 chromosomes. They should have just 2. That's got 3, that's got 3, that's got 1, that's got 1. They're all mutants. They've all got a change in the amount of DNA per cell. This one's got, I've got 2 normal. That's what they should both look like. They're called N. This cell's 2N. It's got 2 chromosomes, 2 homologous pairs, whereas this one's got an extra chromosome, that long one. This one's missing the long one. So how would you describe this image? So let's go for that red box there. How would you describe that? Yeah, what's this black thing here? So this is the spindle fiber, and it's pulling both. Well, it's pulling a chromosome made of chromatids to one of the poles. Okay. And then why is this a non-disjunction? Because the spindle is somehow pulling both homologous pairs of chromosome to one pole. Yeah. This one here should be going to the opposite pole, but no spindle is formed. And I guess this one gets dragged, and then just because of the forces and the kind of like the treacliness of the cytoplasm, it's going to like pull this one across as well. Okay, so yeah, that's a non-disjunction mutation where we don't, where either both chromosomes. I'll just go back to the previous one here. So you can see here, that's what's happened here. No spindle on this side, therefore both chromosomes have gone over to one cell. And on this one, the chromatids haven't separated. So again, spindle didn't form on this side. The chromatids weren't separated like they have here. They've stayed together, and therefore we get 2 copies per cell. And it's still a mutation, but we call it a large-scale mutation. So let's examine 2 genes on the same chromosome. So let's go through these first. So let's look at the diagram. So number 1, what does this circle represent? Yeah, so that's the centromere. That's the centromere that's holding the 2 chromatids together. Okay, number 2, why are these the same allele? Why is this chromatid got the same allele, as in a big A, as the other side? It's identical. Well, this strand has been copied from this strand by semi-conservative replication. This is a chromosome made of identical chromatids. This is a chromosome made of identical chromatids, and they have the same genes at the same loci, therefore they're the same chromosome number, like chromosome pair number 3. However, they differ due to the alleles of genes. Number 3, how many different genes are shown in the diagram? Just checking that the answer doesn't pop up. It's 2. I've got gene A and gene B. There's 2 genes shown. Okay, so that can mess with your head. I've got gene A. Gene A might be the eye colour gene, and gene B might be the tongue rolling gene. How many alleles are shown in the diagram? Right, well, gene A has got 2 alleles, and gene B has got 2 alleles. So I've got 4 alleles in the diagram. What is the genotype of a person with these chromosomes for gene A? So they've got a big A on one chromosome pair and a little a. Okay, the big A has been replicated, but that's kind of irrelevant at this point. So they are big A, little a, and there's a word for that. If you've got a big A, a dominant allele, and a little a recessive allele, they are heterozygous. So I can describe it as a word, the heterozygous for gene A, or I can give their genotype and say they are big A, little a. So there's 2 ways of saying it, saying exactly what it is, or just using a word to describe their genotype. Then 6, what is the genotype of a person with these chromosomes for both genes? And this is what we're going to look at now to kind of apply our knowledge of meiosis to this. Well, if I include both genes, they're big A, little a for the first gene, and the big B, little b for the second gene. They are big A, little a, big B, little b. So I just put both genotypes together as one. Okay. If there was a third gene on, I just add that one on. They are big A, big B, little b, and then big C, if the C's happen to be the same. So this chromosome here doesn't have to have different alleles to this one. I'm just doing it to make it interesting. But this could be big A, and the homologous pair could be big A. And it probably would be if it was a gene like DNA polymerase. Well, we don't have alleles of DNA polymerase. Everyone's got the same DNA polymerase. It's an enzyme. Don't mutate it. It's not going to work. Yeah. So only about 30% of our genes are have alleles. 70% of them don't have alleles. There's just one gene type. Yeah. There's a head sprouting gene. There's one allele. Yeah, there's not a variety of them. If there was a variety of them, it'd lead to like no head being produced. You wouldn't be a viable human. That mutation would be lost from the population in one generation. Good, right. So now we're going to look at how many different gametes if there is no crossing over between these maternal and maternal chromosomes. So remember, in meiosis, the homologous pairs come together and they cross over, but they don't have to cross over. So if these don't cross over, all that's going to happen is that the chromatids are going to, one of each homologous pair goes to each pole. So this one here is going to get pulled to the right as I look at this screen, and this one's going to get pushed to the left as I look at this screen. Okay, and then spindles are going to form that pull the chromatids apart. So therefore, I get a gamete that's got a big A and a big B, another one that's got a big A and a big B, one that's got a little a and a little b, another one that's got a little a and a little b. If these were making sperm, I'd only make two different types of sperm from this chromosome. Yeah, the genotype of the sperm are either big A, big B, or little a, little b. So I can give genotypes of haploid gametes as well, sperm cells and egg cells. Right, so how many different gametes if there is crossing over between these maternal and paternal chromosomes? So if there's crossing over, remember a chromatid on one homologous chromosome is going to lie over the chromatid of the other one, and then they swap equal lengths of DNA. So then I can get my big A and big B, because this one hasn't crossed over. It's just this one here and this one here are going to come together and lie over each other. Called forming a bivalent, and that way the B could have been swapped over, or the A could have swapped over, but it wouldn't have been it wouldn't make any difference. Okay, so I can make 4 different types of gamete with crossing over. I can mix up the A and the B between these chromosomes, I can exchange that B for this B, and therefore I get a gamete that's big A, big B, a gamete that's big A, little b, big B, and little a, little b. I've got more variation. Okay, so it's showing how meiosis can produce greater variation because of crossing over that occurs in the process. So how can we explain these gamete frequencies made by an animal with the genotype that's doubly heterozygous? So I've got lots of big A, big B's, lots of little a, little b's, but I've got not many big A, little b's, and not many little a, big b's. Okay, so the little a, big B, and these two in the centre, I'll stop saying it, it's like a tongue twister. So these two here are rarer than those two there. Okay, and on the previous slide, I said that I can make, here I'm making 4 different types of gametes, and I'm saying this one's common, big A, big B. This one's common, little a, little b. These two are rarer. Okay, these are called all recombinants. These are products of crossing over. Well, they're rarer just because, these are our chromosomes, and crossing over happens, but it's a rare occurrence where the big B and the little b are crossed over. So the gametes big A, little b, and little a, big B are called recombinants, these two, because the alleles have recombined. The crossing over is random and may not happen. So some cells I might just get big A and big B, big A and big B, little a, little b, and little a and little b, and I just, that happens. Occasionally I'll get some crossing over, and therefore I get these recombinants. So a recombinant of genes A and B will only be produced if crossing over takes place between these two genes. Yeah, that's also a good point. I mean, I typed that and I forgot that I typed it, that if they crossed over here instead, and that can happen, well, I'd just end up with a little a and little b on this chromosome, and therefore I'd still get a little a and little b. Yeah, so crossing over has to happen at a locus somewhere between A and B for it to result in a recombinant gamete. Yeah, somewhere around there. Right. Oops. That's bad timing. Often does it happen though, weekly. How can we explain these gamete frequencies made by an animal? Right, so now look what's happened. Now it's these middle two are the common ones, and the top and bottom one are the rare ones. So how can that happen? Yeah, and you can get fixed in your head that no, that can't happen. And well, that can be explained just by this, that maybe the parental gametes, in the homologous pairs, you just happen to on the maternal homologous pair, there's a big A, and it just happens to have the recessive B. So in this case, the recombinants are the big A, big B. To get a big A and big B, I need to cross over between these two here to swap that big B over onto here, and the little b over onto here. So yeah, so those are the recombinants now, the big A, big B, and the little a, little b. These are the common ones here. Make sense? Hopefully does. And we do a lot more of this in year 13, by the way. This is just to kind of get us ready for that, and it's a chance to kind of, if you understand this, you are properly understanding meiosis. So let's just read this through. So crossing over is random and may not happen. Okay, that's why we get lots of big A and little b gametes. Gametes big A, big B, and little a, little b are called recombinants. Yeah, so they've come from being recombined. And this is crucial, the parent still has the genotype big A, big B, little b for these genes. Yeah, it's the same as it was before. On this previous slide, let's go to it. This person's got the genotype big A, big B, little b. Yeah, they've got a big A and a big B on one of their homologous chromosomes and a little a and a little b on the other. and they've still got that here. Yeah, big A, big B, little b. Yeah, it's just that the maternal chromosome has big A, little b alleles, and the paternal chromosome has got little a, big B alleles. Yeah, that's the only reason why I get this difference in frequency. Okay. Again, we do work in year 13 where we breed heterozygotes for 2 gene, and therefore you can look at the offspring and you can work out therefore the positions of alleles on genes by the frequency of the offspring that's produced. Right. So we've gone from that. So as we're going kind of back to it how it was before. Yeah, how can we explain even fewer recombinants? So before, let's go back a bit further. It was 0.44, 0.06. Now it's 0.48, 0.02. Remember, crossing over is random and may not happen. Gametes A, B, and B are called recombinants. Yeah, so yes, the rare ones are the recombinants, so that's correct. A recombinant of genes A and B will only be produced if crossing over takes place between these 2 genes. So the way we get fewer recombinants is to put those genes closer. Yeah, the locus or the loci of genes A and B are very similar now. Yeah, to get a big A, little b gamete, you would have to cross over between these 2, and that's unlikely to happen. If it crossed over at the bottom, that wouldn't affect genes A and B. If it crossed over here, that wouldn't affect it here. It has to go in between them, and therefore they end up being rarer. Interesting. Right. So what is wrong? Extension questions, task. What is wrong about this diagram of a homologous pair of chromosomes? So they're an homologous pair. So what can I say about gene A? Well, it's here on this one, on the identical copy, it's lower down, and genes have a specific locus. Okay, so that's not possible. The gene A has the same locus on both chromosomes. It's in the wrong place. And then what can I say about this one here? BC, not a gene, it would be a copy of it. So I can't just put gene C here because this strand here is a copy of this strand here, and therefore that has to have a B. Okay, or that has to be a C. Right, let's go for this one. Genes A, B, and C are found on the same chromosome. A person has a genotype, therefore. Right, big A, little a, big B, little b, big C, little c. Draw the position of the genes on a homologous pair of chromosomes. Let's look at the data. So big A, big B, big C, they're common. Little a, little b, little c, they're also common. So that gives us the original position of the chromosomes. One, the maternals have got capital letters and the paternals got lowercase letters. Okay, and then A, B, so swapping the B over. Here I've swapped the B over. That happens quite often. Where swapping the C over is really rare. Therefore, again, see if you can draw it out. I think it's something like this. Yeah, that I've got it's big A, big B, big C because they're the common ones, and crossing over doesn't happen every single time. And then to get big A, small b, small c, well, any crossing over in this area here, there's a big area here where that can happen. It can cross over there. So therefore I would cross over B and C would come over to here and vice versa. So that would give me a big A, small b, small c. Yeah, like this one here. Just notice this is incorrect, isn't it? Because it's auto correcting my my things. That should be a capital C. Right. Anyway, I don't want to redo this PowerPoint now, so let's just change it. Let's get it right now. So the rare ones will be big A. That should be big B. Sorry, big B. small c, and little a, little b, big C. Yeah, because it's the chance of crossing over between these two. Let me save that before I'm getting it wrong, because it is the last slide. It was all right until then. Oops. Yeah. So yeah, my big A, big B, big C's are the ones that are going to be made most often. If I cross over between in this area here, I'll swap over the little b and little c, and the big B and the big C. That's correct. And occasionally I will cross over between the B and the C, so I get little a, little b, big C. That's that one at the bottom, and I get big A, big B, small c, because I'm going in between there. So it's now correct. Yeah. Yeah, this would be a hard year 13 question, so don't worry too much. Anyway, that's me done. So again, that would have taken about an hour and a half class with a bit of a break and things. So hopefully that's checked your understanding of some of the difficult topics. Really, we've focused on mutation and meiosis and crossing over and increasing your knowledge of the positions of genes and what happens. Again, I wouldn't worry too much. You know, there'll be a chance to kind of recover next year with there's plenty of workshops and help in biology, so don't worry too much. And send me an email if you don't get anything.